and welcome. FDA has authorized 20 different serological tests, also known as antibody tests, for emergency use during COVID-19. But they have also allowed many other antibody tests to come on market during this emergency. And people have naturally asked the question about their accuracy and reliability. How confident can we be when they give us a positive result or a negative result? How to evaluate the real world performance of these tests. And I've produced a video on that particular topic that you can find on my YouTube channel. But in this particular video, I'm going to address the concept of doing a second test after the first one comes out as positive to probably increase our level of confidence. And this is a recommendation FDA has already made, that under some conditions, a clinician may decide to do a second test. Can we estimate now the improvement in performance? So this is what I'm going to address in this video. FDA has also published an online calculator that you can download. It's an Excel calculator, and I will give you a quick demo of that calculator as well. So let's look into this. First, let's go through the basics very quickly. How do we define accuracy? Let's say we have two sets of samples. One set came from confirmed positive COVID-19 patients, and another set came from confirmed negative COVID-19 patients. So our test can be either positive or negative. So we have four different combinations. If our test is positive for a confirmed COVID patient, it's a true positive. Otherwise, it's a false negative. Or if our test is negative for a confirmed negative COVID-19 patient, it's a true negative. Otherwise, it's a false positive. So let's put some numbers on this. Let's say we test 30 different confirmed positive samples and we find 27 are true positives and 3 are false negatives. Now, on the other hand, we sample 80 confirmed negative samples and we find 76 true negatives and 4 false positives. Now we can calculate the accuracy by sensitivity of 27 over 30, that's 90%, and specificity of 76 over 80, which is 95%. You may also have heard terms like positive agreement, which is same as sensitivity, and negative agreement, which is same as specificity. So now let's look at a scenario where we have about 5% disease prevalence in the population, which means only 5% people have been infected and they had COVID-19. And we test 10,000 individuals with a perfect test. So we'll find 500 true positives and 9,500 true negatives, and there'll be no false negative or false positive. That'll how a perfect test will work. Let's see how our test will work. So we have a 90% sensitivity and 95% specificity. We'll find 450 true positives out of the 500 true positives that a perfect test would catch, and 9,025 true negatives. Rest of them are false negatives and false positives. So is this good enough? How confident are we in our estimate of a positive result, meaning that the patient definitely has antibodies? So for that, we calculate positive predictive value and negative predictive value. In this case, it'll be 48.6%, which is just true positives divided by total positives, which is 925 in this case. So this means that half of the time we are going to be wrong when we measure a positive result on a patient, because we don't know if the patient is COVID-19 patient or not. A priori, we don't have that information. But negative predictive value is pretty good, 99.4%. So this is one example where we may want to do a second confirmatory test only on those patients that tested positive. So let's look into that. For simplicity, we will use the same test, 90% sensitivity and 95% specificity. So we had 450 true positives, 475 false positives, for a total of 925 positive results. Now, when we do a second time, we will find only 405, 90% of 450 as true positives, and we'll find 95% of 475, which is 451 true negatives. So we can build our table again. Here is how it will look like. 405 true positives, 45 false negatives, 451 true negatives, and 24 false positives. This has improved quite a lot because now our positive predictive value has gone up to 94.4%, and negative predictive value has come down a little bit, not by much. So we gave up a little bit, but now we are at close to 91%. So it's fairly good. So as you can see, doing a second test can improve our level of confidence very significantly, right? But this calculation is a little bit cumbersome. I had to spend a little bit of time to figure this out and present to you in a meaningful, easy to understand way. Good news is that we have now a calculator available from the FDA that I will demonstrate how it works. Okay, so this is the Excel worksheet that you can download from FDA's website. It's under the performance of serological tests. So you can download this Excel worksheet. So let's play into that. So let's say our prevalence is, let's say we take a test of 90% sensitivity, 95% specificity, and our disease prevalence, let's pick a high prevalence now, 25%. Just to show you an example. As you can see, this is at 85.7% in high prevalence situations. So this test can work very well in those circumstances, but our COVID-19 prevalence is low. So let's put 5% here. Under that situation, we get only 48.6% like I illustrated in my earlier slides. So it's not good enough. So for simplicity, let's put another 90% test here and a 95% specificity test. And now we are getting 94.5% overall positive predictive value. So it has gone up. What happens if our second test is 99% specific? So we gain on overall positive predictive value by significantly improving our specificity. Okay, so what about if our first test itself is 99% specificity? So we had 82.6% positive predictive value, which is a good number, which means we should pick a test to begin with a high specificity performance. So as you can see that making a selection of the test is very important based on the accuracy information that comes with the characterization of the test. But let's say for some reason you end up picking a test which is not very high, not even, let's say 95% specificity. So you may consider a second test, but which second test to pick? From a clinical point of view, make sure we understand that all these tests work differently. So they target different antigens, different antibodies, and we need to be able to understand how they work, not only looking at their specificity, sensitivity numbers. It'll be better to pick a test which works very differently. And in that scenario, even if you pick a test with 95% specificity second time, you will end up getting a pretty high level of confidence in the positive result. So there's a lot to it than might appear on the surface, but the good news is that you have these tools available to play around and understand how those numbers will turn out in the real world. So I will be producing a lot more videos on these topics or any other topics that you might be interested in. Just let me know, send me a note through my LinkedIn profile or through my website, and I'll be very happy to produce new videos for you. But the best way to keep in touch and track all these developments is to subscribe to my YouTube channel, and I'll show you how to do that. You can go to my YouTube channel and subscribe, or you can follow me on LinkedIn where I post these videos, and I also give you links to come back to my YouTube channel for more videos. Like I said, send me your questions and comments or other topics of interest, and I'll consider that for future videos. Finally, thank you for your interest and attention, and I hope all of you are staying safe.